When you think of animal research, what comes to mind? I asked some of my friends to see what they thought.
Rats or mice.
Small animals like fish.
The research that is done, like cancer research, that is actually very useful. Mice research.
Monkeys.
I kind of think of lizards because of like Spider-Man.
While mice, rats, and primates are popular subjects, by the end of this segment, I'm hoping you'll have a new animal to add up your sleeve. Organisms have been used to model human health as early as the mid-1800s, when Gregor Mendel used pea plants to better understand inheritance. The pea plant was one of our earliest model organisms, or species that act as simplified representations of humans. Namely, through systems, genetics, or behavior, they can advance our understanding of biology. Discoveries about DNA replication and cell division are largely thanks to the humble fruit fly, while Arabidopsis, a mustard plant, has contributed significantly to our understanding of circadian clocks and proteins. These model organisms, among others, have directly led to improved treatments and advancements in human health and agriculture. So, with so many models out there, how do you choose which one to use? This decision ultimately comes down to a few boiling points: what you're studying, costs, and generation time, or the time between an organism being conceived and being able to reproduce. For example, plants function on a much larger timeline than animals. Just think about how long trees take to grow. If you wanted to study plant development, using something fast-growing like corn would be much more efficient. Animals like fruit flies produce extremely quickly and are cheap to maintain, but as invertebrates have key genetic differences to humans. It's important to note that while there are pros and cons to every model organism, at the end of the day, they're still models. Different aspects of human biology can be best represented by different species, and context is key. While mice and monkeys may be some of the first model organisms to come to mind, I wanted to zoom in on an animal making a splash in science: Danny Orario, more commonly known as the zebrafish. These fish are native to the freshwater rivers of South Asia, identifiable by deep blue stripes that trace the fish head to tail. Only growing to be a couple inches long, these hardy fish can live in a wide range of salinity, pH, temperatures, and elevations. So, how did these fish get from shallow riverbeds to the lab? Although there are other fish models, namely Medica, carp, and goldfish, none have had such prolific success as the zebrafish. Charles Creaser had proposed zebrafish for embryology research as early as 1934, but it gained traction in the 1960s when George Streisinger at the University of Oregon started using this organism.
How is it related to humans? It's a fish. We would think that there's not a lot of similarities.
How much of fish research is done to learn about fish, and how much of it is used to learn about other species?
What y'all testing on them?
How would that even remotely translate to human beings?
The zebrafish has many appealing features. Let me give you the elevator pitch. They're vertebrates, small in size, use external fertilization in transparent eggs, develop rapidly, are genetically accessible, and have characterized behaviors. Let's dive into some of the fine details. These fish are no spineless creatures. They're vertebrates, so many features overlap with human biology, particularly at the genetic and molecular level. Evolutionarily, humans and fish shared a common ancestor about 400 million years ago. Fish are quite infamous for their poor memory, but even when looking at the brain, you might be surprised to hear that there are regions that are functionally homologous, meaning that although they're not the exact same, they serve a similar purpose. I mentioned earlier that these fish only grow to be about 2 inches long. When they're larvae, a popular experimental age, they are 4 to 5 millimeters long, easily fitting under microscopes and in pretty much any experimental setup you can dream up. In terms of maintaining the fish, keeping aquariums is space efficient, and fish care is simple. Fish also lay eggs all year round in hundreds, allowing for studies with large sample sizes. This is especially beneficial for drug trial tests since you can get a large data set in a small amount of time. These fish don't make much of a fuss, especially compared to mice or other mammals, which require more space and expenses to maintain. A big reason zebrafish were proposed as a model organism in the first place is that they use external fertilization, meaning the eggs are laid outside of the fish before hatching. These transparent eggs allow for a direct window into their rapid development. The hallmarks of cell division are well characterized in the 48-hour period before hatching, down to the number of cells present every hour. This accessibility as an embryo is a major advantage zebrafish have over other vertebrate models like mice. Looking at a brief timeline of development, you can see why this is an area in which zebrafish excel. Fish lay eggs overnight, hatch 2 days after fertilization, and by just 5 days old, all major organs have already developed. Since you only have to wait about a week for fish to be an appropriate age for most experiments, there's a pretty quick turnaround. By 3 months old, they reach sexual maturity and are ready to breed the next generation of fish. At the turn of the century, scientists began exploring the possibility of sequencing full genomes, or the complete genetic code, of model organisms. Zebrafish are no exception to the buzz. After successfully sequencing simpler creatures, scientists tackled the human genome, which was fully sequenced in 2003. Zebrafish, which by now were gaining popularity, followed suit in 2013, unlocking novel possibilities for research in genetics and development. Zebrafish have a genetic equivalent, or ortholog, for about 70% of genes in humans. For reference, chimps have about 96% similarity, mice 85%, and fruit flies 60%, making zebrafish a competitive model for human biology. With the genome unlocked, the doors for genetic accessibility were now wide open. Creating different strains of fish to fit a variety of experimental conditions is increasingly easier as technology improves. Previous studies have used these fish to model diseases like osteoporosis, leukemia, and autism spectrum disorders. Zebrafish are also currently the only genetically accessible model for appendage and heart regeneration. I'm going to say that again. Their hearts and fins, when damaged, can literally grow back, which is pretty crazy. When genetic accessibility and genome editing technology collide, new research methods become available. We can now breed zebrafish that lack pigment and genetically insert proteins that glow under a specific wavelength of light, such as green fluorescing proteins, or GFP. This transparency and successful genetic alteration gives zebrafish an edge in recording how the brain responds to stimuli. Proteins label calcium, which is an indicator of neural activity. You can imagine putting a transparent fish under a microscope that is then focused on a region of the brain you're interested in, then shining a light over the region to see which neurons glow during a given stimulus. The more neural activity present, the brighter the neuron will glow. These recordings make beautifully comprehensible videos where you can see waves of neural activity. To compare to mice again, getting the same neural recording would require surgically removing a part of the skull to gain access to the brain, while in larval zebrafish, you can do this non-invasively. That's pretty cool. While zebrafish are a strong contender for studying the brain, they also have a library of characterized behaviors that can be used during experiments. For example, a startle response is marked by a C-turn, where the fish will bend their body into a C shape and turn around. Sounds and vibrations cause a startle response, easily replicated in a lab setting with speakers. Zebrafish are also social creatures. They exhibit shoaling, which is when a group of fish swim in the same direction as a pack. I like to think of the reflective fish forming arrows in Finding Nemo. The optomotor response, also known as the OMR, is a phenomenon where fish follow the direction of visual motion as a form of stabilization. In the lab, this visual motion, or flow, can be simulated by moving black and white stripes. You can think of it as sort of an optical illusion. When the stripes move, the fish perceive it as the world moving around them. This is a method Dr. David Ehrlich's lab at the University of Wisconsin-Madison utilizes quite a bit. In this lab, I've been researching the OMR alongside PhD candidate Nick Milicic, who I brought in to talk a little bit more about it.
The OMR is like this really strong, powerful, reliable stimulus for like getting the fish to swim and turn. Our kind of understanding of the OMR is limited because we basically only ever do it in the horizontal plane. And if you think about the OMR as like this stabilizing reflex, if they're getting like pushed around by the currents, then they're gonna get displaced like vertically as well as horizontally. So we were thinking, okay, is there a vertical OMR response essentially? We basically got this like crazy setup where we put the fish under a microscope and like we can see into their brains because they're transparent, and we can basically play a vertical OMR stimulus for them while also like giving them a flick. Basically look in their brains and see how everything wires together and activates with different pairs of visual motion and vestibular sensation. Hopefully that will one day like be informative in like understanding how the circuitry that helps you catch yourself when you walk works, so we can look in humans for where it fails.
Could you explain a little bit about what fish can teach us about the motor system specifically?
So zebrafish can teach us about the motor system in the sense that they have a lot of the same neural circuitry as us. Of course, their bodies are very different, but a lot of the underlying circuitry is the same, and the way it's wired is the same. A lot of their brain regions are like highly conserved evolutionarily. And so even if like their, I guess, like biomechanics aren't directly translatable, a lot of how everything is organized and how experience shapes those connections is very valuable.
How can fish movement be translated to how humans move? Like, what's the connection between that?
I don't think humans have a direct optomotor response, but we do have similar reflexes because the optomotor response is evolved in response to optic flow, which is like whole field visual motion, right? Like your whole world is moving in one way or another, right? And so we wouldn't necessarily respond in the same way as a zebrafish because, we're obviously not identical, but a lot of the underlying baseline circuitry is probably the same. Optic flow can be very disorienting. Like if you've ever like stood next to a train or something that's really close, that can like disorient you and throw you off balance. So we can learn about like how we respond to optic flow in terms of like what areas of our brain are active, and particularly like how the vestibular system, our balance system, might react to the sense of the entire world moving in a direction outside of your control.
Let's assume our hypothesis that this vertical optomotor response does exist. Mm-hmm. What's next and what are some future directions?
There's maybe an instinct to ask, who cares how fish swim? But like as I've talked about, understanding how fish swim is important for like establishing a baseline to like design experiments off, and those experiments can eventually lead to benefits for people. I think basic research it's like kind of hard to talk about, right? Because it is abstract, right? And you're absolutely right. Like a lot of basic research stuff won't necessarily like translate to any clinical success for years or even decades after its use. Right. But the work is still important because you never know what might be that critical last piece that leads to a breakthrough, right? There's like countless cases of that sort of thing happening throughout history.
Right. Yeah, experiments inform other experiments, and like it's a totally like butterfly effect sort of thing.
Yeah, absolutely.
So, although there might be a little bit of bias, there's some pretty cool research happening with zebrafish. While there are clear upsides to this model organism, again, there's no model without its limitations. Of course, the fish brain is eons more simplified than that of humans. Since they aren't mammals, there are key differences in major organs between the species, namely the respiratory and reproductive systems. Furthermore, all behavior tests that can be performed in zebrafish have equivalents in mice, while some behaviors in mice don't translate to zebrafish at all. While zebrafish are strong models for drug trial tests, fish of course live in water, so testing water-soluble drugs proves to be difficult as well. Do these limitations mean that we shouldn't use zebrafish in research at all? Of course not, as there are still so many positives. No model is perfect. It's important, however, when designing experiments that these limitations are kept in mind and accounted for. There's also an ethical side to animal research that has been in the public eye since the start. Is raising animals in a lab for research ethical? Does the good outweigh the bad?
Feels a little bit wrong, but on the other hand, it's like a necessary evil, because without that, how could you progress and make people feel better?
It doesn't bother me at all. It's not good or bad. I don't care.
I'm fine with it. It's science. You have to research it.
I have a mixed feeling because I don't believe in cruelty.
I know that to even get animal research going, you need to go through an ethics board.
There are strict protocols and guidelines animal researchers must follow, mandated by federal law. In a zebrafish lab, all members are required to be trained in ethical animal handling and experimentation. Here at University of Wisconsin-Madison, regular inspections of lab conduct ensure that all activities are in accordance with, quote, federal regulations, veterinarian standards of care, campus policies, and facility standard operating procedures. If something doesn't meet these regulations, then the lab has to adjust or face being shut down. But can there be a future of research without animals? In vitro testing, meaning experiments that take place outside of a living organism, such as in a petri dish or test tube, has been growing in popularity. Stem cells are cells that rapidly multiply and often have the ability to turn into other cell types, a leading alternative to model organisms. While stem cell research shows much promise for treating neurodegenerative disorders like Alzheimer's and Parkinson's disease, storing stem cells is costly. Furthermore, cells outside of the human body function differently from those in a living subject. There are still many aspects of human biology that are better fit to be studied in living model organisms. With the wave of artificial intelligence, AI models can predict and track trends of massive data with efficiency and speed. If we could accurately model organisms through a simulation, then animals would not have to be used at all, and the cost of animal maintenance could be saved. Another powerful tool is neuroimaging, where scans like fMRI, PET, and CT measure neural activity in living human subjects. While the landscape of model organisms used in research is ever-changing, there has been clear progress in science and health resulting from zebrafish research. They have helped identify potential drug treatments for leukemia and melanoma, uncover the genetic workings of Alzheimer's and Parkinson's, and identify genes associated with heart defects at birth, among other discoveries. Zebrafish are undeniably causing ripples in science. In the last 10 years alone, the NIH has published almost 128,000 articles citing zebrafish. As we uncover the mysteries of these fish, that number will only continue to grow, furthering our knowledge of neuroscience, behavior, and biology. With that, I hope you leave this podcast with a new favorite model organism. So the next time someone asks you, what animals come to mind when you think of animal research? Say zebrafish. Can't get enough of zebrafish? Me neither. You can find the full list of citations used to make this podcast below. I want to give a special thank you to all my friends who chimed in on this podcast, particularly Nick, as well as my PI, Dr. Ehrlich, for teaching me everything there is to be excited about zebrafish.